Pancreas, it lies parallel and it lies beneath the stomach. It has got three parts: a head, body, and tail, and it has both an endocrine as well as an exocrine gland. Now it consists of three type of cells: the acinar cells, the centroacinar cells, and the ductal cells. The acinar cell is responsible for the secretion of pancreatic juice, which is rich in enzyme. The centroacinar cells, again it rises from the junction of acinus and it grows into the lumen of acinus. And the ductal cells is responsible for producing watery pancreatic juice, which is rich in bicarbonate. So acinar cells is responsible for secretion of pancreatic juice rich in enzyme, whereas ductal cells is responsible for watery pancreatic juice which is rich in bicarbonate. Now coming to the pancreatic ducts. Now various ducts, the secretions from the various acini, they join finally to form the ducts. There are mainly a main pancreatic duct as well as the accessory pancreatic ducts. Now both main as well as the accessory pancreatic duct will drain into the second part of duodenum through separate opening. Your main pancreatic duct, also known as pancreatic duct of Wirsung, will join to the common bile duct to form the ampulla of water, and this ampulla of water is guarded by the sphincter of Foley. Similarly, your accessory pancreatic duct, also known as duct of Santorini, enters the duodenum. Now we learn about the composition of pancreatic juice. Pancreatic juice is a colorless, clear, watery fluid. It is secreted around 2 to 2.5 liters per day. It is alkaline in nature because of high concentration of bicarbonate and has a specific gravity of 1.007 to 1.042. Coming, it is composed of 99.5% of water and 0.5% of solid. Now again, these solids can have either organic and inorganic constituents. The inorganic constituents include water, sodium, potassium, calcium, magnesium, bicarbonate, chloride, and sulfate. Whereas organic constituent, it includes the enzymes. It contains various enzymes as well as organic substances. The various enzymes are trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase A and B, colipase, pancreatic lipase, pancreatic amylase, RNase, DNase, and trypsin inhibitor. And other inorganic substances like albumin, globulin, kallikrein, lysosomal enzymes, alkaline phosphatase. Now coming to the enzymes in pancreatic secretion, it's a very important question. Now the enzymes, as we have already learned, it is produced by the acinar cells, and they mainly act only in alkaline medium. And there are four main enzymes which is secreted by the pancreas. First one is your protein splitting enzyme, you have carbohydrate splitting enzyme, fat splitting enzyme, nucleic acid splitting enzymes. Now the protein splitting enzyme include trypsin, chymotrypsin A and B, proelastase, carboxypeptidase A and B. Now the feature of all these enzymes are, these enzymes are in the active form and only they will be activated once they are secreted into the intestinal sac, or else in the pancreas they will be in the inactive form, and the optimal pH for the action of this enzyme is around 8 to 9. Now first we learn about trypsin. The trypsin is the most powerful proteolytic enzyme of pancreatic juice. It is normally secreted in an inactive form, that is trypsinogen. Now this trypsinogen will be converted into an active form trypsin by an enzyme called an enteropeptidase or enterokinase. And what is this enteropeptidase or enterokinase? It is an enzyme which is secreted by an duodenal mucosa. So you can see that the inactive trypsinogen gets converted into trypsin by enterokinase or an enteropeptidase, which is an enzyme secreted by duodenal mucosa. And once your active trypsin is formed, it can further activate trypsinogen by autocatalytic action. Clear? So So what are the actions of trypsin? It mainly converts protein into proteases and polypeptides. It coagulates blood. It helps in the curdling of milk. It activates other enzymes like chymotrypsinogen, procarboxypeptidase, proelastase into active form. It activates phospholipase A2, and function of this phospholipase A2 is it converts fatty acid to lysolecithin, and also it has got autocatalytic action. What do you mean by autocatalytic action? It catalyzes a further conversion of trypsinogen into active trypsin. Now coming to the second enzyme, that is chymotrypsin. Chymotrypsin, again it is secreted in an inactive form, that is chymotrypsinogen, and this is activated to converted into an active chymotrypsin by trypsin. And the main function of this chymotrypsin is it helps in the coagulation of milk, as well as it hydrolyzes proteins into small polypeptides. Coming to the third enzyme, elastase. Again, elastase is secreted as proelastase, which is an inactive form, and it is converted into an active form, that is elastase by trypsin. And the main function of elastase is it again digests elastin. And the last enzyme is carboxypeptidase. The carboxypeptidase is secreted by, again it's mainly secreted in an active form called as procarboxypeptidase, and it is activated by trypsin to active form carboxypeptide. So this is four enzymes, which is a protein splitting enzyme. So which are the four protein splitting enzymes? They are trypsin, chymotrypsin, proelastase, and carboxypeptidase A and B. Now the next one is you have the carbohydrate splitting enzyme, mainly pancreatic amylase. A pancreatic amylase is it's secreted in the active form, and the main function of pancreatic amylases is it hydrolyzes starch, glycogen, and other carbohydrate to form the disaccharides. And normally the plasma amylase level will become only high in case of acute pancreatitis. So it is one of the tests which is used to find out whether the person to know whether a person is having an acute pancreatitis. Now coming to the fat splitting enzyme. Your fat splitting enzyme include the pancreatic lipase, colipase, the phospholipase A2, bile salt activated lipase. Now coming to the fat splitting enzyme, pancreatic lipase, it is a powerful lipolytic enzyme, and it mainly hydrolyzes fat to glycerol esters and fatty acid. Now coming to colipase, it again acts as an cofactor for pancreatic lipase, and thus helps in the activation of pancreatic lipase. That is, colipase is necessary for the activation of pancreatic lipase. Coming to phospholipase A2, it is normally secreted in an inactive form, that is phospholipase A2, and the trypsin, as we have already mentioned, the trypsin is mainly responsible for converting this into its active form. And what is the action of phospholipase? Is it acts on the phospholipids to form the lysolecithin, which will damage the cell membrane. In acute pancreatitis, your phospholipase A2 is activated inside the pancreatic duct, and lysolecithin formation will occur, and due to which there will be disruption of the pancreatic tissues. Coming to the last one, that is a bile salt activated lipase. It mainly catalyzes hydrolysis of cholesterol esters of fat soluble vitamin, phospholipids, and triglyceride. The last one is the nucleic acid splitting enzyme, which include the ribonuclease and deoxyribonuclease, which mainly hydrolyze nucleic acid releasing nucleotides. Then you have something called as a trypsin inhibitor, which is mainly synthesized and stored in the acinar cells that secrete your trypsin. Normally, it prevents the activation of a trypsin in the acinar cells and in the ducts. But whenever there is a damage of pancreas, or whenever the ducts get blocked, a large amount of secretion gets pooled in the damaged area of pancreas, and under this the effect of trypsin inhibitor is more, and due to which it will cause a activation of proteolytic enzyme, which results in the digestion of pancreas within few hours, and this condition is called as pancreatitis. Okay, so what happens is, normally your trypsin inhibitors prevent the action of trypsin in the acinar cells, but whenever there is a damage of pancreas, what happens, your the effect of trypsin inhibitor gets overwhelmed, and due to which there will be excessive activation of your proteolytic enzyme, which will result in the digestion of pancreas, and that is called as autodigestion of pancreas, and this condition is called as acute pancreatitis, which can be fatal. So I'll stop here. So in the first class you have to know, today I taught you about the various enzymes which is in the produced by the cells, SNR cells. There are four type of enzyme, that is protein splitting enzyme, carbohydrate splitting enzyme, fat splitting enzyme, and nucleic acid splitting enzyme. Protein splitting enzymes are trypsin, chymotrypsin A and B, proelastase, carboxypeptidase A and B. Then you have carbohydrate splitting enzyme, that is pancreatic amylase. It's the only amylatic enzyme which is present in your pancreatic juice. Then you have your fat splitting enzyme, which includes the pancreatic lipase, colipase, phospholipase A2, and bile salt activated lipase. Then you have your nucleic acid splitting enzyme, which include the ribonuclease and deoxyribonuclease. And then you should know the function of trypsin inhibitor. Thank you.